The polygonal model corresponds to a proposal for a very simplified representation of biomolecules compared to the classic Fisher projection. Let's see how the glycolytic pathway is shown by this model. Glucose metabolization in this pathway has 10 reactions. In the first reaction, glucose is phosphorylated to glucose 6-phosphate at the expense of ATP. This product isomerizes to fructose 6-phosphate, that is phosphorylated to fructose 1,6-bisphosphate, also at the expense of ATP. After this step, the molecule splits into 3 carbon compounds involving several steps that we will not describe in detail here. Even with these small 3 and 6 carbon molecules, there are many details that are hard to remember. The number of atoms visualized is close to 220. Given the importance of phosphate in glycolysis, the phosphate group can be shown in color, making it easier to see what is happening. Another interesting way to show glycolysis is to use a colored circle containing the letter P to symbolize the phosphate group, PO32-. This notation is often used to enhance visualization of the glycolytic pathway. If this substitution simplifies the visualization of this pathway, then why not extend it to other chemical groups? For this, we can consider hydroxyls, carbonyls, and carboxyls. For example, the hydroxyl group may be represented by a triangle. In this case, we can also consider hydrogen-substituted hydroxyls that occur in phosphate groups. Carbonyls correspond to aldehyde or ketone groups and may be represented by squares. Carboxyls, on the other hand, may be represented by circles. The remaining carbons that are part of the biomolecule skeletons, as well as the respective attached hydrogens, can be omitted without any harm to the final representation of the biomolecules, since we know that the carbons have a total of 4 covalent bonds. We can also place the non-asymmetric carbon bonds in an inclined position, while keeping the other bonds horizontal. The approaches described here allow the construction of a variety of very different representations of the original molecule. Such representations may increase the possibility of remembering the structures of biomolecules that participate in metabolic pathways, in addition to providing more compact and graphic forms of these components.